Everyone, today in this video I will tell you about monoclonal antibodies. So let's start our topic. Monoclonal antibodies. Monoclonal. Mono means single and clonal means clone. In a simple term, monoclonal antibodies refers to monospecific antibodies that are made from identical immune cell. Okay, in simple term we can say that monoclonal antibodies are monospecific antibodies that are made from identical immune cell, means from the same immune cell. Monoclonal is written as small m, capital A, small b, or it is also written as MOAB. You can use any one of this derivative, any one of this abbreviation. Okay. Now, these monoclonal antibodies, they are monovalent. Means they bind to only one specific epitope. What is epitope? It is a part of the antigen which bind to the antibodies. Okay. Antibodies are proteins and they are synthesized in blood against specific antigen. Okay. Antibodies are the protein which are synthesized in blood against a specific antigen. Antibodies can be mass produced by scientists. Okay. These are called monoclonal antibodies because they are produced from clones of the sample lymphocyte. From starting I'll tell you, monoclonal, it is a single clone. In simple terms, it refers to the monoclonal specific antibodies that are made from identical immune cells. Abbreviation for monoclonal antibodies, small m, capital A, B, or MOAB. These are monovalent. They bind with only a specific antigenic epitope. Antibodies are protein which is synthesized in blood against specific antigens. and the scientists produce antigenic mass. These are called monoclonal antibodies because they are produced from the clone of the sample lymphocyte. I'll tell you. See, this is a B lymphocyte cell and this is the antibody. The next one is, this is another B lymphoid side and these are the antibodies. Okay, these are from different clone. Now these are from same clone. These are the B lymphocyte cells and these are the antibodies. Okay, another will take, these are the B lymphocyte cell and this is antibody. Antibody, a Y-shaped structure, that is why I am writing here Y. What is this indicate? This indicate that antibodies produced are identical and they all target the same specific antigen. How it can be taken? They are collected from blood serum of an animal. Okay, and whatever we are obtained from blood serum of an animal, that antibodies are heterogeneous, means they all are different and contain mixture of antibodies. Therefore, these antibodies or whatever we are getting the sample that do not have characteristic of specificity. Means we can't use this type of antibodies because they are not specific. So to get a specific antibodies, if a specific lymphocyte after isolation and culture in vitro, now they are become capable of producing single type of antibodies, which means they can bear specific against specific antigen, and this is known as monoclonal antibodies. Okay, I'll tell you in a short way what we have to do, from where we can collect this antibody, monoclonal antibody. First we have to collect the blood serum of an element. that blood serum of an animal, whatever we are getting, that are heterogeneous and it contain the mixture of antibodies. That antibodies or that mixture do not have a characteristic of specificity, means that and that mixture contain the different kind of antibodies which can bind to a different antigen. We don't need here different kind of antibodies. We are targeted a specific antigens. For that we need a specific antibodies. For that what we have to do, we have to take the lymphocyte for the isolation, and after culture in a vitro, that become a single type of antibodies, which means that have a specificity against the specific antigen, and these type of antibodies are known as monoclonal antibodies. Okay, I think you understood now. Now the next is production of antibodies. Production of monoclonal antibodies. To produce antibodies that target a specific antigen of our interest, we need to obtain correct lymphocyte. For production of monoclonal antibodies, first we have to produce antibodies that target a specific antigen. Whatever our interest of antigen, we need to produce the antibodies against it. First we have to produce that. For that we need to a correct lymphocyte should be obtained. What it means is, see, this is our antibody. Okay, we need to produce a same. This is a B lymphocyte and this contain antibodies. And the another type is, this is B lymphocyte and this contain these purple color of antibodies. So here first we have to obtain specific antigen, means we have to produce these type of antibodies. This is different, these are same. So first we have to obtain this one. This is achieved by injecting genetically modified mice with this antigen. Okay, here we are using a mice for getting our antibodies. I'll take this as mice. We have to inject here and we are getting these type of antibodies. Okay. Now their immune response, there means the mice, its immune response produce B lymphocyte that makes antibodies of our interest that bind to antigens that binds to specific antigen. Now we have to collect these cells. After collecting, we have one problem for further process. What is that problem is the difficulty is here lies in making of the clones of this cell. Means whatever we are getting the cell, it's difficult to getting a clone because these lymphocyte do not divide very easily in culture. Because of that, we are getting difficulty in making the clones of same cell. To get overcome from this type of problem, next scientists think and they develop, they said cancer cells we can use. The cancer cells are used in lab because these cancer cells, they divide very easily This is the cancer cell and this can easily divide in a culture. This is a Petri plate which contain a specific medium and these are the cancer cells. These all are divide. Okay. Now what the scientists now did, scientists fuse a mouse B lymphocyte with cancer cell from bone marrow to make a particular cell, and this is known as hybridoma. What they did is they took this B lymphocyte of mouse and fuse with cancer cell. This is cancer cell and this is B lymphocyte of mice. Okay. After the fusion, they get the cell that is known as hybridoma. Now these cells are then cloned to make lots of identical hybridoma. This is a Petri plate which contains the specific medium and here hybridoma cells were obtained. Okay. The antibodies produced by these cells are collected and purified. From these cells we are getting the monoclonal antibodies from hybridoma cell. We are getting these monoclonal antibodies. In a brief, I'll tell you from the starting. For the production of monoclonal antibodies, to produce a particular antibodies for a specific antigen of our interest, we need to obtain the correct lymphocytes. For this, we have to inject the genetically modified mice with the antigens. The immune system of that mice will produce the B lymphocyte that make antibodies that bind with the targeted antibodies. After that, collect all cells which are produced from the B lymphocytes of mice. Here we are getting a problem. This problem is we can't make the clones of these cells because lymphocyte does not divide very easily in the culture. To overcome this problem, scientists use the cancer cells. The cancer cells, they are used in laboratory because they divide very easily. This is the cancer cell, this is divided. Now what they did is they fused B lymphocyte of mice with the cancer cell. They get the hybridoma cells. These are the hybridoma cells and which produce anti-monoclonal antibodies. Now these cells, whatever we are getting previously, we are cloned to make a lot of identical hybridoma. We make identical hybridoma, and this will produce the monoclonal antibodies. That's all. Thank you for watching my video.